exploration of protein synthesis.
As told by
Kyle and Rachel.
Now let's look at a seal. Let's look at a seal's cell and in the nucleus of the cell.
Protein synthesis.
Let's examine the molecules involved in this process.
DNA.
Deoxyribose nucleic acid has a double helix, a deoxyribose sugar, and a phosphate group. It also has 4 nitrogenous bases. They include adenine, thymine, guanine, and cytosine.
mRNA.
This messenger RNA differs from DNA in that it is single-stranded, it has a ribose sugar, and instead of thymine, it contains uracil as its 4th nitrogenous base.
Its function is to carry the message for making proteins from the nucleus to the cytoplasm. tRNA.
Transfer RNA has a specific amino acid attachment site. It also has an anticodon. Anticodons are usually written in the 3 to 5' direction, opposite of the mRNA codons, which are in the 5 to 3' direction.
Its function is to carry amino acids to ribosomes during translation. Ribosome.
A ribosome contains 2 subunits, the large top unit and the small bottom unit. 60% of this is rRNA, or ribosomal RNA, and 40% is protein.
There are 3 tRNA binding sites. The P site, which holds the tRNA carrying the growing polypeptide chain, the A site, which holds the tRNA carrying the next amino acid for the chain, and the E site, which the tRNA leaves the ribosome here.
This is the primary site for protein synthesis.
The first step is transcription, in which RNA is transcribed from a DNA template.
After transcription is completed, mRNA processing occurs.
The final stage in protein synthesis is called translation.
Initiation is the first step in translation. The small ribosomal subunit binds to the mRNA molecule. The initiator, tRNA, with the anticodon UAC and the amino acid methionine base pairs with the start codon AUG. The large ribosomal subunit binds to the smaller subunit, completing the complex. The initiator, tRNA, is in the P site. The energy here is provided by GTP.
Elongation is the second step in translation. GTP and the elongation factor's hydrolysis provides energy. The first process of elongation is codon recognition. Incoming amino-acyl tRNA binds to the codons in the A site. Second process is called peptide bond formation. A peptide bond is formed between the new amino acid and the carboxyl end of the growing polypeptide, previous amino acids. The ribosome catalyzes the bond. The third process in elongation is called translocation. The A site tRNA shifts to the P site, taking the mRNA strand along too. The mRNA shifts its position by one codon. Simultaneously, the P site tRNA moves to the E site and is released from the ribosome.
Termination is the last step in translation. The ribosome reaches a termination codon on the strand of mRNA that signals for translation to stop. The stop codons are UAA, UAG, and UGA. The A site accepts a protein called a release factor. This causes the addition of a water molecule to the polypeptide chain. It hydrolyzes the completed polypeptide chain from the P site tRNA, thus releasing it from the ribosome. The complex then breaks down into the mRNA strand, the two ribosomal subunits, and the polypeptide chain.
Thanks for watching our video. Bye.
At Philadelphia University, to study the effects of poaching, biology student Kendra Talbot didn't hang out in class. She hung out here, in Kenya, conducting research that explored the delicate balance between human and wildlife survival. To change the world, you have to be in it. At least that's our philosophy. Check out Kendra's and other Power to Do stories on our website.